Hi everyone, my name is Corinne and today I'll be talking to you about immunity and antibody production. The essential idea for this topic is based on recognition of some cells and destruction of foreign materials. How does this recognition work, you might ask? Well, it all comes down to the plasma membrane. So plasma membranes have little structures called major histocompatibility complex, or MHC glycoproteins. As you can guess by the name, these structures are made of, well, protein. They can be found in all nucleated cells. What's special about these molecules is that they help with cell recognition. So, for example, all of my cells have the same MHC glycoprotein. As my cells encounter each other in my body, they recognize my glycoprotein themselves, and that's why they don't attack my own cells. However, if the cells aren't recognizable, leukocytes or white blood cells set off an immune response. And this brings us to our first definitions. So antigen is the name we use for unfamiliar surface molecules on a pathogen. These antigens are how our immune system classifies cells it encounters. Some pathogens include bacteria, virus, fungi, and protozoa. When leukocytes find antigens, they set off an immune response. Nonspecific immunity is when a phagocyte destroys any cell it recognizes as foreign using endocytosis. Specific immunity is when antibodies are created to destroy pathogens. Both of these responses are necessary in order for someone to be considered immune to a disease. So, now that we've gone over some definitions, let's talk about how immunity actually works. So, this right here is a pathogen. It has entered someone's body and is now looking to infect some cells. But before it can do that, a phagocyte approaches. So here's my little phagocyte for you guys. So this special type of leukocyte engulfs the pathogen and destroys almost all of it. However, the antigen is preserved. So I'm going to go ahead and take off these antigens. And then the phagocyte destroys this. Now what's going to happen is these antigens are going to be presented to inactive helper T cells by the phagocyte. So let me go ahead and do that for you guys. When the inactive helper T cell encounters these antigens, they become activated. This allows them to find inactive B cells, which I'll bring in right here. And then this is another type of lymphocyte, by the way, and it exposes them to the antigens. See how it fits perfectly? That's how you know that this is the proper B cell for this. So when the antigen matches these receptors, this B cell becomes activated. Activated B cells begin to clone themselves via mitosis. Their clones are designated to become either plasma cells, and I have a few of these right here, or they can become memory cells. I have a few of these right here. So plasma cells produce and secrete antibodies that aid in the destruction of pathogens. Remember, these are what our pathogens look like. And they do this because their specific shape actually matches up with the pathogens, as you can see right here. So these Y-shaped proteins have four major functions: neutralization, opsonization, agglutination, and complement activation. Neutralization is what allows antibodies to prevent intruders from entering other cells. Opsonization is when they mark pathogens to make them identifiable for other cells. Agglutination is the process of attacking and clumping foreign material together to make it easier for pathogens to be destroyed. And complement activation is when antibodies encourage other immune system components to attack the pathogen. Once these antibodies have been secreted, they begin to stick to antigens. So I'm going to go ahead and move them right here. And you see how the shape lines up with the antigen. That's very important for this process to work. Then the next thing that happens is macrophages, or big eaters in Greek, are alerted to the presence of a pathogen because of how many antibodies are in the way. So here is my little macrophage. Much like regular phagocytic cells, it engulfs the invader via phagocytosis. However, unlike before, all parts of the pathogen are destroyed, including the antigen. So up to this point, this entire process can be classified as primary immunity. But remember those memory cells from earlier? Well, they play a part in secondary immunity. These memory cells remain in the body long after infection. If the same pathogen returns, they are responsible for secreting antibodies to mark them to be destroyed by macrophages. So it's very much like it was earlier. These antibodies are going to go ahead and connect to the antigen. This macrophage is going to find it, and then it's going to destroy the pathogen. This immune response is much quicker and usually causes less severe symptoms than the first time. This process is known as secondary immunity. In fact, secondary immunity is what causes vaccines to be so successful. In order for someone to be considered immune to a disease or illness, memory cells must be present to fight off infection. Well, that's all for now, folks. Thank you so much for listening. I hope you enjoyed this, and I especially hope you learned something new in this. Bye!